Good afternoon, everyone. Thanks for coming to my presentation. My research is about the Xylem ion loading and osmotic stress tolerance as the determinants of differential of salinity tolerance in barley and wheat. This is the background. Soil salinization is a global issue that affects plants' growth and limits agricultural production. Currently, 20% of the world's cultivated lands, which is equal to 6% of the world's total area, is threatened by salinity. But in Australia, 2/3 of the agricultural area is facing the potential of transient salinity. So screening and breeding for salt-tolerant genotypes is becoming an urgent task. The first chart shows the diversity in salt tolerance in various species, which increases in short dry matter after growth in solution or sand cultivar containing sodium chloride, which lasts at least 3 weeks, and the relative to plant growth in the absence of sodium chloride. But we can see that the barley here is much more tolerant than wheat. And plant growth responds to salinity in 2 phases. One is the rapid osmotic phase that inhibits the growth of young leaves, and another one is slower and ionic phase that accelerates the senescence of mature leaves. The physiological and molecular mechanisms of tolerance to osmotic and ionic components of salinity stress are reviewed normally at cellular, organ, or the whole plant level. This graph shows the physiological changes in the cellular level. We can see that the biochemical functions associated with tolerance to plant salt stress. The schematic presentation of a plant cell includes 3 compartments, which are defined by the plasma memory and the tonoplast. So they are normally including the selective accumulation or the exclusion of ions, or the control of ions uptake by roots and transporting to leaves, and some like the induction of the antioxidative enzymes and the plant hormones. So that's the normal changes, physiological changes in the cellular level. We choose some physiological contrasts for our work, like the Xylem loading, including the sodium and potassium and water relation, and the osmotic adjustment, such as the inorganic ions or the compatible solutes. So last one is the balancing transpiration and the carbon dioxide oxygen assimilation. So that includes the stomach density and the stomach conductance. This is my research proposal. 50 barley and 50 wheat, which is totally 100 genotypes, were selected and treated with sodium chloride. And we, through the determination of these physiologic traits, we will do the statistical analysis. Normally, they correlate with biomass or green yield, or the plant survival, like the leaf injury. And the statistical analysis is normally including the regression analysis, linear correlation analysis, and the principal component analysis, or the cluster analysis. And also, we will choose some salt tolerant and salt sensitive genotypes for the further study, like the kinetics of xylem ion loading or the mechanism of xylem loading using the MIFE measurement. So the blue part, were already finished, and what I'm doing now is the red part. It's like the data analysis and the kinetics of xylem ion loading. And the green part is my future work. This is a photo of the glass house. The left one is the plants under treatment under the salinity. The right one are the control ones. The following slides are my current results, but just some of them because it's too many. I just choose some of the results of different physiological traits of 50 barleys. So we can see that the significant difference exists in among the 50 barleys, like the stomach conductance, osmolality, sodium content in leaves, and the potassium content in xylems. Then I did the correlation analysis and the regression analysis, and I found that the osmolality, the osmolality, sodium content in leaves, potassium, sodium content in xylem, and the potassium content in xylems have a significant correlation with dry weight. Sorry, the dry weight is one of the biomass. So this is some part of the results of 50 weights, and still the same as the barley, like the GS. Sodium content in leaf, potassium content in xylems has a significant difference among these 50 weights. And then the correlation analysis and the regression analysis. From this, we found that both potassium content and the sodium content have the obvious correlation with the plant survival. Summary. There is significant difference in each physiological trait among 50 barleys and 50 weights. The concentration of sodium and potassium in leaf obviously correlates with plant survival. It's the leaf injury, and the biomass and the green yield. Correlating salinity, stress tolerance with control of xylem ion loading is obvious. So generally speaking, different mechanisms of the salinity tolerance exist in different genotypes. So what I'm going to do next is has three parts. The first one is the further data analysis, like the principal component analysis or the cluster analysis. The second one is the further study about the mechanisms of xylem ion loading using MIFE or the patch clamp. Yeah, depends on my supervisor. And the third one is the effect of potassium availability of xylem ion composition. Because we found very interesting results. Before, like the potassium in is much higher than normal ones, because the plants is like growing in the spatial pot mix, really high. So we like to do the study about this. About the mechanisms of xylem ion loading, we have two parts. One part is electric physiology. The other is study of the genome expression. According to the previous results, we will choose here 4 barley and 4 weights to study the mechanisms of the xylem ion loading using the MIFE measurements on the stellar tissue, which is treated by the sodium chloride, mannitol, or the hydrogen peroxide. To study which factors are regulating the xylem ion loading, we were choosing the 2 constructing barley genotypes and treat them with the signaling factor like the ABA, calcium, or the pH, and so on. And yeah, we will determine the potassium, sodium, and the chlorophyll. Sorry, it's not chloride. It's the chloride flux using the MIF. Hopefully, I will do some further study about the iron transporters. Here are some transporters here. This is the SOS1 and the HKT, which is located on the cell Xylem apoplast interface. In activation of these iron transporters, will help to elucidate the function of these transporters and the pattern of Xylem iron loading. At last, I'd like to thank all the people who ever helped me since I came to Australia. And really, I want to thank my supervisors, Sergey, Misha, Lana, and all the lab mates. Thank you.